Yeah, it's my absolute pleasure to be back here virtually, if not in person, to tell you about some ongoing research in my lab by folks in my lab group. And I would love to, yeah, just dive right into it. Before I start, so the title of my talk is Some Answers and More Questions about the Role of Small Particles in Ecosystems. This is work in progress, but it'll be, I think, a nice update on what all we've been up to. I'd like to thank my co-conspirators. So these three graduate students have done a lot of the work that I'll be talking about today. Lauren Sullivan, who's graduated, Kate Perkins, and Dylan White. Kate's going to be graduating very soon. And then also two other folks who've sort of got roped into some of this work, Dr. Manuel Montano and Frank von der Kammer, Dr. Frank von der Kammer from University of Vienna. I'd also like to thank my funding sources, National Science Foundation, Montana Water Center, and the Institute on Ecosystems. The National Science Foundation, of course, funded the UN Bridges program as well as the state EPSCoR. All right, so I also want to, and I won't read off everyone's names, but I also want to thank a variety of folks, graduates, undergraduates, volunteers, technicians, and faculty members and others who have contributed this work, whether they realize how much they've contributed or not, they have. So if your name's on this list, fantastic. If I've forgotten you, feel free to shoot me an email afterwards and read me out. I apologize. I tried to be inclusive. And really, by training, I'm an ecosystem ecologist, and I am really interested in the ways that that the world around us works. I'm interested in a couple of very broad overarching questions. First question is, what are the drivers and mechanisms of element cycling? And the second is, how do chemical perturbations, how does chemical change in the environment change element cycling? And really here, I'm most interested in a range of different chemical perturbations. They can be things like salt water coming into coastal freshwater wetlands, or it can be nitrogen from fossil fuel emissions and agriculture raining into forests. But the work that I'm going to tell you about today is focused on other chemical perturbations. It really sort of got started with work during my postdoc that was focused on looking at engineered nanomaterials and their impacts on ecosystems. So things like titanium dioxide, which are sometimes used as a pigment on donuts, but also used for more noble purposes, such as serving as a photocatalyst for water treatment. Carbon nanotubes, which are the next one down, which can be used to make stronger, more resilient tennis rackets, but also can be used for making thread that has the conductive properties of copper, but the strength of steel, all at much lower weights. And then also quantum dots, which are small particles that, depending on how big they are, you can tune the color that they fluoresce, and you can use them to make some pretty vibrant television. So that's a quantum dot television. And so back in the 2000s, early 2000s, we saw a very massive rise in the use of these small particles in a lot of different consumer products. And when I say small particles, nanoparticles are very small. They're 1 to 100 nanometers. And if we look at, I've got sort of a size scale here that I like. We've got nanoparticles down here at the bottom end, 1 to 100 nanometers. We've got colloidal particles, which overlap. And in fact, it was really the bottom end of the colloidal particle size range that nanoparticles were carved out of. And then we have clay. We have pretty good overlap there too. And then we have bigger particles: silt, sand, clay, gravel, cobble, boulders. And when I started working on this work, which is not going to be the focus of my talk today, but it's an important part of the backstory, I got really interested in the fact that we oftentimes discretize the world into being dissolved and things that are not dissolved. And so things that we operationally define as being dissolved, there are things that if we take a filter like this, and we can use a variety of different size cutoffs, 0.7 microns, 0.45, 0.22, depends on your discipline and how much water you're filtering. And we confront that with a water sample, push that water sample through, we have this idea, make this assumption that what passes through is these small ions, and that all the particles are retained. And furthermore, we assume that based on that, the particles that are retained, those tend to be less bioavailable. They tend to be unstable when they're in aquatic environments. They tend to just fall out. Whereas the dissolved fraction, this operationally defined fraction, we assume it's dissolved solutes and that they tend to be pretty bioavailable. But if you look at the overlap here, and so I've chosen my favorite particle size or my favorite filter size, you'll see that it overlaps with nanoparticles, the majority of colloidal particles, and a lot of the distribution of clay particles. And so dissolved, this operational definition, it's not really dissolved. It's filtered, but it's not necessarily these small dissolved ions. And that's important. That's an important distinction. So when we think about submicron particles, so that's colloidal particles and nanoparticles, they have some interesting properties because of their size. So they have this high surface area to volume ratio. As they get smaller and smaller, as we go from right to left here, that ratio just skyrockets. And that makes them very reactive and gives them some unique properties, as we talked about earlier with respect to those different consumer products. They tend to become fairly stable in suspension. So the rate at which they settle ends up being smaller than the rate at which they move via Brownian motion, just random walks as they get smaller and smaller. And then finally, they serve as an efficient delivery system. So much like a giant wooden horse is a good way to get soldiers into your enemy's base, the idea is that these small particles can be a way to get something that's a potential resource, or in some medical applications, they use it as a way to get antibodies certain places, or a way to get chemotherapy agents to certain places. It can be a really effective way of delivering things. And so some of the early questions in our research are, will organisms take these particles up? And do they cycle biogeochemically as other elements do out in the environment? And if so, how? And will they have effects on organisms? And so in our early work and work of others, so I'll start actually with work of others, some of the work as we were getting started, they would add massive amounts of nanoparticles to the food, feed it to a protist like this Tetrahymena, and find that sure enough, you'd end up with all these food vacuoles full of nanoparticles, and they'd stop moving very quickly, probably partly because they were full and partly because they're full of cadmium selenide, which is a bit toxic. But from our own research here using silver nanoparticles, which are an antimicrobial, used commonly as an antimicrobial, we found that looking at sort of the base of the food web, sediment and paraphyton, even though we were adding concentrations that were somewhere down here on this logarithmic scale, we'd see they accumulate in the sediment and the paraphyton in these chronic exposures. And then we'd see high concentrations in snails and clams. As we go to a variety of other invertebrates and onto the vertebrate mosquito fish here, we'd see lower concentrations, but still much elevated versus their environmental concentrations. And then we'd also even see it transfer trophically from the aquatic to the terrestrial compartment and into spiders. Here is visualizing some of that uptake. Here we used duckweed, very small aquatic plants, added either silver nitrate, zero valent silver nanoparticles, so just metallic silver, or silver sulfide nanoparticles. And we saw that all of these were taken up either over 24 hours or over 60 hours. And these are particles. And certainly with the silver sulfide, it's not likely that these were being disassembled, because when we looked at the speciation of the particles, they were still silver sulfide. So we see uptake of these particles. We also saw extensive evidence that they're cycled biogeochemically. I'll just run through this really fast. So again, with silver, we see that zero valent silver, elemental silver, was transformed in the environment to silver sulfide. This is the same process that you'll see if you have any silver, anything that tarnishes. Those silver nanoparticles, either as zero valent or silver sulfide, can oxidize, releasing silver ions. And those silver ions in turn can also be retransformed back into either silver sulfide nanoparticles or zero valent silver particles in the environment. We also found evidence that we'd see silver ions bound on metal oxide particle surfaces. So here we have titanium dioxide particles, either from biosolids or from soil. We're not sure exactly which, because it was an experiment where we had both. And also they'll bind with organic matter via thiol groups, so sulfur connected to carbon and some other stuff. And this range of transformations changes how bioavailable and how toxic that silver is, but it's strong evidence that these things are cycling biogeochemically. We also saw evidence of, even at realistic exposures, we would see some ecosystem level effects. So here is an experiment where we added wastewater treatment biosolids with either silver nanoparticles or with silver nitrate as sort of our positive control. And we saw more of an effect on microbial biomass. So think of, the scales with the amount of microbes in the soil. We saw a decline in microbial biomass versus just the slurry alone. And we saw an increase in N2O production versus just the slurry alone. And it was stronger than that silver nitrate treatment, which actually we made as a strong positive control. It was 4 times the concentration of the silver nanoparticles. So you see a pretty robust signal here. But just because it's statistically significant doesn't really mean that it is all that ecologically important. And really, these were fairly small signals at the low concentrations of silver at which we had it, and persisted for a short period of time. But this really got me to thinking a lot about the cycling, accumulation, and impacts that we're seeing from these manufactured nanoparticles, and got me thinking back to my roots, which in part are in soils, and thinking about the full range of natural small nanoparticles. And there's also particles that we create incidentally. And in reality, these manufactured particles are kind of the rare thing out in the environment, in ecosystems. And it's really more the incidental and the natural particles that are much more dominant, especially the natural particles. But it got us to thinking, well, why wouldn't they also be cycled? They probably are. And sure enough, in looking at some of our data, some of our experiments, we saw some interesting patterns emerge. So here we have silica nanoparticles in the roots of alfalfa that was grown hydroponically, but hydroponically in rock wool, which is basically expanded basalt. Interestingly, these silica nanoparticles were in the plant, and they were actually clustered around silver nanoparticles that we added, which is intriguing. We can't say definitively whether this was by happenstance or an adaptive feature of these plants to try and detoxify the silver, but it was intriguing at least. We also, in looking at some aquatic plants, while we were expecting to see either gold particles or silver particles in some wetland plants that were growing under the surface of the water, we saw titanium dioxide, and it looked like that titanium dioxide was sort of nestled in among the mesophyll elements in the plant. That was fairly unexpected, and this is just from soil. There's about 1% titanium dioxide in soil, and it stands to reason that's probably where it came from. In looking at a coal ash spill, we went upstream a little bit and saw a seep from that coal ash basin, and found evidence of these arsenic-coated iron nanoparticles. Boy, doesn't that sound delicious. And then we also, going downstream and actually looking in the zone of the spill, we found titanium dioxide particles which were coated in copper. And so this got us thinking about, well, what are some of the potential roles? We can sort of extrapolate from what we've seen from these engineered materials. What do we expect we might see? Well, they could serve as a subsidy. So to the extent that they contain some element that a plant or a microbe or an animal needs, that could be a subsidy. It could be they are a stressor. Copper-coated titanium dioxide particles doesn't sound great to me. Sounds less bad than arsenic-coated iron particles, but still. And so this has sort of motivated the research questions that I'm going to talk to you about today, which are sort of the first steps along this process. And so these research questions are to what extent are different elements found in different size fractions, and to what extent are these size fractions bioavailable? To what extent are they interacting with organisms, maybe taken up by organisms? And I would like to tell you this story in sort of as 2 separate projects that I think come together to give a broader understanding of this. One was focused on wastewater lagoons, and one was focused on a mine waste contaminated stream. And if you're thinking, I wonder if that's the Clark Fork, sure enough it is. And in fact, the wastewater lagoons are in the Clark Fork watershed as well. In So I think for a lot of you, probably most of you, I don't need to give too much of a breakdown on the Upper Clark Fork, but just in case. The Upper Clark Fork has this extensive history of mining and processing of copper, deposition of waste on the floodplains and into streams, redistribution of that flood in a series of floods, big one in 1908 and some others that followed. And it has resulted in contamination of the stream channel and floodplains with arsenic, cadmium, copper, lead, and zinc, and associated iron and manganese. And so, you know, to orient folks, in case you haven't seen this map or don't know where you are or where we are, so here we've got Butte up here in the Berkeley pit. Here I'm hiding in a basement over here. And the area in between, it varies in contamination, but when that material moved through, there was contamination seen. You know, Missoula is 200 kilometers downstream. It was seen 400 or more kilometers downstream. I want to plug in another event tonight. If folks haven't heard about it, there's an Upper Clark Fork River Superfund update put on by the Clark Fork Coalition and others. The link for it is tinyurl.com/UCF for Upper Clark Fork, and then SFund for Superfund. And I'd highly recommend, if you can't get enough of the Clark Fork, I would recommend checking that out. All right, so wastewater lagoons are the most common wastewater treatment method used worldwide. And in fact, in Montana, 84% of our municipalities use lagoons. They're fairly low maintenance, they're fairly low cost, although many of them are in non-compliance, and it's okay that you can't read these. In fact, I prefer you can't read which ones have been in non-compliance in the past 5 years, but there are many that have been. I was approached by a post-bac student who's interested in doing a master's and ended up doing her master's with me and is now working for the Standards Division of Montana Department of Environmental Quality. And she was really interested in better understanding the movement of metals through these systems. And you know, we talked about these overarching research questions, and she thought that was pretty exciting. And we came up with a project where we would ask this question of, you know, what's the distribution of elements among different size fractions, with a focus on that sort of colloidal size fraction. And she did this by sampling 6 different wastewater lagoon systems in Western Montana. And she also conducted an experiment to look at to what extent are these different forms bioavailable, and she did this through a simulated lagoon experiment. And the method that we used is pretty low tech. We just took water samples from the field, brought them back to the lab, and then fractionated them with filtration. So instead of just 2 fractions, we did 3 fractions. We did whole water, which is just taking an aliquot of the water intact. We filtered it through, in this case, a 450 nanometer filter, and then also through a 1 nanometer filter. And so that gives us these different size fractions, or filter fractions, which we then converted into different size fractions after analyzing the samples using ICP-MS, and we focused on aluminum, arsenic, cadmium, copper, iron, manganese, lead, and zinc. We calculated these 3 different size fractions. So suspended particulate matter, which here, because of the size of the filter, we're defining as larger than 450 nanometers. Basically, it's just bigger than the filter. We did that by taking our whole and subtracting our filtered water. We looked at colloidal particles, which is, in this case, 1 to 450 nanometers. We did that by taking less than 450 nanometer water, our filtered water, and subtracting the ultra-filtered. So we put it in special tubes and put it in a centrifuge, and it pushes the sample through that 1 nanometer filter. We wouldn't want to do that by hand. And then we looked at the truly dissolved fraction, which is just that sample that's gone through that ultra-filter. So what do we see? Well, I'm going to focus on colloidal particles here. And so, you know, the question, to what extent are these different elements found in different size fractions? Let's focus on the colloidal fraction, and specifically looking at what percentage of the filtered water consists of colloidal particles. And so what we see is that for copper, iron, manganese, lead, and zinc, we see that colloidal particles can be over 50% of what's found in that filtered water. And for aluminum, arsenic, and our bonus element here, chromium, we see that even though they're pretty low component of the colloidal fraction, or their fraction in the colloidal fraction is pretty low, is still between 12 and 25%. So it's small, but it's not nothing. Based on these data and based on our interest in looking at what happens to these materials in wastewater treatment lagoons, and also from a perspective of thinking of these as sort of a point source, and therefore there's a potential for increasing their efficiency, we conducted an experiment looking at the potential use of floating treatment wetlands to try and remove some of those metals and see to what extent it removes the different size fractions. Floating treatment wetlands, if you're not familiar, you've got sort of a fibrous and floaty mat with plants growing hydroponically. The roots grow through it, and you are able to just set them loose. It's pretty passive, don't have to manage a lot unless you have a lot of shorebirds trying to land in them. And what we sort of anticipated as sort of our conceptual model of how this system would work is that these suspended particulates would come in, and they're big, and a lot of them are big enough that they're just going to settle out. Dissolved solutes, truly dissolved solutes, so smaller than 1 nanometer, we assume that they might be available for biological uptake, either by the roots themselves or by periphyton, by this mixture of algae, bacteria, and even fungi that grows on those roots. We also assumed that we might see some entrapment of the suspended particulate matter on that periphyton, and that material might ultimately slough off over time. There's going to be some turnover expected, and therefore that may contribute to the sediment. But a real question was, all right, well, so we expect that. We don't really know for sure what to expect with colloidal particles. Are they going to behave somewhere in between the suspended particulate matter or dissolved? That was sort of our guess, but we didn't really know. In order to test these floating treatment wetlands, we constructed 12 small wastewater lagoons. Lauren went and got wastewater from a local wastewater treatment lagoon system with a very friendly operator, and brought it back and set up 6 high concentration, so 2-to-1 ratio of wastewater to groundwater, and 6 low concentration, so to 1-to-2 ratio of wastewater to groundwater. And then in half of them, illustrated here with the green rounded squares in the middle, we had these colonized floating treatment wetlands. And you can see they're planted with a variety of different plants. So many of these are metal accumulating plants, and we figured that was probably a good direction to go. I'm going to show a few different figures here to represent these data. And so we've got some where we're using what's called the log response ratio. And basically what this does is it takes the ratio of the mean concentration we see in the floating treatment wetland treatment, so the treatment, versus our control, which is open, no treatment wetland. And basically, due to the properties of natural logs, it's the same as subtracting one natural log from the other. And so if it's negative, that means the concentration was lower with the floating treatment wetlands. If it's positive, that means it's higher, which would be not such a good thing. And so we looked at all of our different metals and arsenic. So what do we see? Well, we saw that floating treatment wetlands, they decrease metalloid concentrations at high wastewater concentration. They also do it at low wastewater concentration, but for simplicity, I'll just show you the one. For all of the elements we looked at, maybe with the exception of arsenic. Arsenic, just in general, appears not to play well with others, or with floating treatment wetlands at least. The floating treatment wetlands also, most interestingly to us, changed the size distribution of the metalloids. And so this is at the end, well, really of the metals. This is at the end of the experiment. And you can see that for the suspended particulate matter, we have a couple that are significantly lower, as represented by no overlap with this line. Some that seem to be lower, but especially for the colloidal particles, we see very clear patterns where the floating treatment wetlands facilitate the removal of these particles. And surprisingly, or counterintuitively to me, for the truly dissolved fraction, it really didn't change that much. There wasn't much of a difference, and there wasn't that much removal. So that was somewhat interesting. Of course, arsenic is up here being recalcitrant. So we know that it's removing these colloidal particles, but we don't know really what they are. We don't know what the particle types are or the sizes. We know the range is somewhere between 1 to 450 nanometers. It could be that we have So here we have our hypothetical metal M+, could be sorbed on metal oxide particles. It could be sorbed by or bound by high molecular weight organic matter. It could be existing as its own particle. It could form its own nanoparticle. It could be that it's bound up more intimately with iron oxide, forming this whatever mineral that is. And it could be that we have sort of a core shell arrangement. So we might have iron oxide in the center, we might have something else on the outside. And it's kind of beyond the scope of our work thus far to totally resolve this, but this is something that we're pushing after. And I'll show you some data where we can sort of discriminate between this fate on the left and these fates on the right. So in summary, in wastewater lagoons, we found that copper, iron, manganese, lead, zinc, over 50% of that filtered fraction was colloidal. And then for arsenic, chromium, copper, it was much lower, 12 to 25% of that filtered fraction was colloidal. But still, that's an appreciable amount. So if you think about exposure of organisms to these elements, and if you're only thinking about the truly dissolved forms, you might be kind of missing what's happening. Now, it might be that the particles have a protective effect, or it may be that they serve as this Trojan horse, they serve as this effective way of delivering it to these organisms. So to what extent are they bioavailable? Well, the flowing treatment wetlands removed metals, and the dominant fate was in the periphyte and sediment. And really we saw that the removal of that colloidal fraction was larger than of the suspended particulate matter or the truly dissolved solids. So that was a little bit unexpected and pretty interesting. Shifting gears now and shifting to talking about a mine waste contaminated stream, namely the Upper Clark Fork River. This is work mostly of my master student, Kate Perkins, who's going to be finishing up by the end of the semester, which is really exciting, and my PhD student, new PhD student, Dylan White. And our work in the Upper Clark Fork is made possible by two dominant funding sources, one through the NSF through the LTREB program, and one through the NSF through the EPSCoR program. And that's the CRUISE project for the Consortium for Research on Environmental Water Systems. We routinely measure either monthly or bi-weekly the Clark Fork, as well as doing a variety of other activities out there. And we monitor it from basically just below the Warm Springs Ponds all the way to Missoula. 13 different total measuring sites. And if we look at the concentration, this is just going to be in terms of total recoverable metals, so that's like that whole fraction from the wastewater work, or in the filtered fraction, so that's what goes through, in this case, a 700 nanometer filter, a little bit larger filter. What we see is that there's a lot of total recoverable metals that aren't captured by this filtered fraction, and so that would be that suspended particulate matter. But let's, rather than look at this, let's sort of disentangle it. I also, on several of these figures, I'll have these letters down here. They just refer to some sites that folks may or may not know: Warm Springs, Deer Lodge, Gold Creek, Bear Gulch, and University of Montana, just by campus here. And these are roughly spaced out every 40, 50 kilometers along the river. So when we size fractionate the water, and we've got our suspended particulate matter in purple, our colloidal in green, and our truly dissolved in sort of this mustard yellow, what we see is that, well, we kind of see a few different patterns here. So for aluminum and iron, we see that, okay, the suspended particulate matter dominates, but the next dominant fraction, and the dominant fraction in that filtered component that we historically have called dissolved, is colloidal particles. And that's an interesting result. When we then move forward to looking at manganese and copper, we see there that we see a similar amount of truly dissolved manganese and copper to the suspended particulate matter, and the colloidal fraction is smaller. But out of that dissolved fraction, out of that filtered fraction, it still represents between 10 and 30% of that fraction. So again, from the perspective of thinking about exposure, it's probably important to think about it, especially for copper. And then from the perspective of arsenic, it seems like it's mostly truly dissolved. I'm pretty convinced here that neither the suspended particulate matter or colloidal particle fraction are as important. Maybe around 10% colloidal, but it's pretty consistently at that level. Now let's think a little bit. This is averaged over the time period from January of 2019 to September of 2019. Let's think about the patterns here across time. And of course, snowmelt hydrology drives a lot of patterns, both over space, as we see increasing discharge as we go from Warm Springs up to just above Missoula, and then time as we look from January through October. We see this snowmelt increase in the summertime, and then it declines in winter, or in the fall, late summer, fall. And I've put these drops here to represent the start of snowmelt runoff, or the big snowmelt runoff, roughly the peak and then the end. And I've got that on these contour plots, which are coming up next. The other thing I wanted to mention is that there is a period here in the data where we see ice, and maybe we see some signature of that in these contour plots. So I've switched the axes here. We've got suspended particulate matter, colloidal, and truly dissolved forms here from upstream to downstream. And the first thing to notice with iron is, again, that colloidal fraction is sort of intermediate between suspended particulate and truly dissolved, but it's definitely much higher than the truly dissolved fraction. And you can also see that really the peak concentration seems to be as the flow starts coming up and at peak concentration. And that seems to be the case for the most part across all three different size fractions. We do see something interesting going on up here a little bit earlier in the season. And whether that has anything to do with ice, probably not, but it's an intriguing pattern there. I'm not going to show every single element, but I'll show copper at least. And what we see with copper is that suspended particulate matter, colloidal, and truly dissolved all have sort of that peak around peak discharge, right? Also here a little bit different, we see that the truly dissolved is larger than the colloidal, which aligns with what we saw when we average across time. But interestingly, interesting from my perspective, in the colloidal fraction, we have this early peak, sort of diffuse and sort of rolling out a little bit differently up and downstream. And then we also see this late season high colloidal copper. And I don't know if that's due to productivity, changes in pH that facilitate the formation of particulate forms of copper or what, but pretty intriguing. So we see sort of peak concentrations as flow increases, and then in SPM and truly dissolved, and then the colloidal maxima seems to have one that's in the spring, and it's a little later and continues into the fall. So where are we going from here? And that's work that I'm not going to talk about today other than to allude to it. This is future work by Dylan White, PhD student working with me. We're going to move from our qualitative understanding to examining the drivers. Obviously, discharge is important, but past work has shown, at least on daily time steps, temperature is important, pH can be important, dissolved organic matter likely is important, sediment metals, productivity of algae and aquatic plants is also likely important, if for no other reason than it drives some of these other factors, and conductivity may play a factor as well. So to summarize this portion, what we see in the Upper Clark Fork in terms of looking at the distribution of elements in different size fractions, well, colloidal, aluminum, and iron dominate in the filter fraction. We're not necessarily concerned about those as contaminants per se, but it's interesting, and you'll see why in a little bit. Colloidal, manganese, and copper are also important in that filtered fraction, maybe 10 to 30% of that filtered fraction. So again, when thinking about exposures, especially to copper, that may be important. And colloidal arsenic, relatively unimportant, 10% or less, but there still is a colloidal component. So for some organisms, that may be important. If they're, say, filter feeders, they might experience that more. Also, discharge seems to be important as a driver across these different size fractions, and I would argue biogeochemistry likely is too. We'll know more as we start diving into those results a little bit more. Now, we still don't know the particle types or sizes. The range is a little bit larger here, 1 to 700 nanometers, but we still don't know this. And so as a little bonus, I wanted to just briefly tell folks about a side project that became Kate's main project, trying to look at how big are these particles and what is their composition. And this is collaboration with Dr. Manuel Montano now at Western Washington University in Bellingham, but formerly a postdoc with me at Duke, and then we were both postdocs there, and formerly a postdoc with Dr. Frank von der Kalmer at the University of Vienna. And this uses a technique that, for any folks who were at Jamie Leeds' presentation, you may have seen this. It's ICP with a time of flight mass spectrometer, and spelling it out all the way, we used a single particle inductively coupled plasma time of flight mass spectrometry. I can't even say it, or a single particle ICP TOF-MS. And single particle ICP-MS has been around, but the time of flight element is comparatively new. The basic idea is that you introduce your sample, and these small dots are ions, and the big ones are colloidal or nanoparticles. They enter into the plasma, they're introduced into the instrument. In the plasma, they're dried out, and the particles are broken up and ionized, and then they come through. And when you have what used to be a particle, you get a cloud, and when you have little ions, you get individual ions. And so what you see in this data here is that you see the background signal, which is those ions, and then you see these peaks. Those peaks correspond to particles, and the size of the peaks actually corresponds to how many moles of a given element are in that particle. The technique basically, I'll sort of zoom through. The particles enter into the time of flight instrument, the mass analyzer. They're separated based on their size, and so you can resolve for an individual particle the different components of that particle. Let me skip ahead. Basically, you can look at that data, zoom in on it, and you'll see peaks that have multiple different elements in those peaks. So we looked at the Upper Clark Fork using this approach. Kate took a set of samples in September of 2018. There were a total of 23,000 and change particles detected, 144 distinct particle types. And one thing that was really interesting is that the number of elements, and this is from a subset of the sites on the Clark Fork, just to a little bit past Garrison. And basically, the higher the number of elements in those particles, the fewer particles that were detected. So here I've got the top 20% of particles by just the relative abundance of the particles. And that represents, out of the total number of particles, 99% of the total particles by number. We've got single particles indicated by an individual element by itself. We've got two element particles with two different elements, and the order of those elements confers information about the relative abundance of the two elements in those particles. And we have some three element particles. Here's just two of them highlighted. First thing to note is that iron and manganese are the most common in particles. The second thing to note is that single element particles are more abundant than two or three element particles. And then if we look at these multi-element particles, we find that iron is in almost all of those multi-element particles, with the exception of this manganese set of manganese lead particles there. Now we can dive into the data a little bit more, and this looks a little overwhelming, but I'll walk you through it. So these are basically plots that show the distribution of number of atomoles per particle. We've got our single element particles, two element and three element particles grouped together by the elements in the particle. And the thing to notice here, iron, in terms of the number of atomoles, tends to be greater than most elements, with the exception of maybe titanium and silica in particles. Nearly all of the multi-element particles, again, contain iron. One thing that's interesting is we can also look at the ratio of these elements to one another, and the average ratio for iron and manganese is pretty much equimolar, a little bit more manganese than iron. That's different than what we see, for example, for iron and copper here, where we've got a ratio of 1 to 0.2, or iron and lead, 1 to 0.1, or in these multi-element particles where manganese and iron or iron are more abundant than the other components of those particles. Looking at particle size, we see that most of these particles are either down in the nanoparticle range, and again, iron particles are, I think, they're a huge percentage, maybe 60 or 70% of the total number of particles, and the majority of them are in the nanoparticle range. Certainly for manganese, we see the same. And for those other particles, many of the other particles that are larger, they're still in the colloidal size range, many of them in the small colloidal size range. We also see that as we increase the number of elements within a particle, that particle size on average is increasing. So they get bigger the more complex they are. Now, is that because the particles, you can't detect them until they get to a certain size? I might be inclined to guess they are, in part. So you'll see that a lot of these distributions here on these violin plots are pretty flat on the bottom, and that suggests that we're not capturing the smallest particles within those. We can also look at the loads of colloidal particles going downstream. Basic pattern here is they increase as we go downstream. So this is the amount moving past a point in the river per unit time. So for iron, it's grams per second moving by. For some of these other components, it's smaller, but it's appreciable. And even for arsenic, we're seeing milligrams per second moving by in the river. Also of interest is that for some elements like manganese, we see this leveling off, which was a bit surprising. We also see evidence of increases following a tributary, where that tributary is not really putting in a whole lot of zinc, but we see change in the form of zinc and the formation of particles or release from the bed sediments. We also see evidence of decreases in terms of arsenic and lead. So these particles are dynamic. This is evidence that they are cycling at some level. So to break this all down before getting to some conclusions, our single particle work reveals that we see iron, manganese, and manganese iron colloidal particles are dominant. We see nanoparticles and small colloidal particles throughout the river, increasing in concentration as we go downstream. But one caveat is that the single particle ISP TOF-MS technique, it needs a certain amount of an element to detect it. So it might be that on a lot of that iron, manganese, and manganese iron, on a lot of those particles, we may have some level of copper and arsenic and the like absorbed on there. And we actually expect that, but it may not be sufficient for us to be able to detect it, while as with some of the larger particles, we're actually able to. Okay, so some take-home points. I would argue that dissolved is not dissolved, and that's okay. And filtered samples are okay to call dissolved for some ions. You know, sodium and potassium are fine, but certainly for iron, it's not terribly accurate. Submicron particles are ubiquitous and they can be bioavailable. We know this from both our work with manufactured nanomaterials as well as these incidental and natural nanoparticles. We see that these natural and incidental particles are potentially contaminant vectors. So we see that from those multi-element particles where you have iron, for example, with lead or copper or what have you. And because of that, they could potentially be serving as stressors, as might those single element particles consisting of copper or arsenic or lead or what have you. But of course, like research, any good research project, we've got a lot more questions. So how rapidly are these colloids and nanoparticles cycled or spiraled through the stream? How do the different size fractions drive movement into and through organisms and in food webs? And, you know, we're actively pursuing answers to these questions, and hopefully in another 4 years I'll have some, well, actually hopefully in another 2 or 3 years I'll have some good answers to those questions as well. And so with that, I'll thank you for your time, and I'll stop talking.